Over 20 years, melatonin use among U.S. adults has increased from 0.4 to 2.1%. Millions consume these supplements as a general-purpose sleep aid. This contradicts the American Academy of Sleep Medicine's clinical practice guideline for chronic insomnia. The task force explicitly recommends that clinicians avoid using melatonin as a treatment for sleep onset or sleep maintenance insomnia in adults. Clinical trials show that for adult primary insomnia, melatonin reduces the time it takes to fall asleep by an average of only 11.7 minutes. This is often insufficient to provide a meaningful clinical benefit. For chronic insomnia, guidelines favor targeted GABA-A receptor modulators, medications like azopiclone or zolpidem, which provide robust sedation and sleep maintenance. Public perception and evidence-based medicine have drifted into a state of misalignment. While the supplement is a household name for sleep, objective data for neurotypical adults remains weak. The clinical failure of melatonin as a general hypnotic requires a reevaluation of its actual physiological role in the human body. This confusion is compounded by regulatory status. While melatonin is a prescription-only medication in the EU and UK, it is sold in the US as an unregulated dietary supplement. Testing of commercial U.S. products reveals that actual melatonin content ranges from 83% less to 478% more than what is stated on the label. Purity is also an issue. 26% of tested supplements were found to be contaminated with unlisted serotonin. This lack of oversight makes consistent dosing impossible for the average consumer and complicates clinical evaluation by physicians. You cannot accurately measure the efficacy of a molecule if the dosage being administered is a moving, contaminated target. Oral melatonin faces significant biological barriers. Bioavailability is highly variable, with only 10 to 56% of the dose reaching systemic circulation. After ingestion, plasma levels show a sharp spike, reaching a peak concentration in approximately 60 minutes. This is followed by rapid clearance in the liver, where the cytochrome P450 enzyme CYP1A2 hydroxylates the molecule for excretion. Extrahepatic metabolism by CYP1B1 in the brain and large intestine further accelerates systemic clearance. Sustained sleep maintenance requires a stable plasma concentration. Melatonin's natural kinetic profile is a rapid spike followed by a crash, making it ill-suited for keeping an adult asleep throughout the night. Common substances can also alter this curve. Fluvoxamine inhibits metabolism and increases concentrations, while smoking and alcohol consumption decrease them. Because of its aggressive clearance and low bioavailability, oral melatonin is a highly inefficient tool for sustaining sleep in neurotypical adults. Despite failures in the general adult population, melatonin demonstrates statistically significant efficacy in specific clinical cohorts. In children with autism spectrum disorder and ADHD, studies show a decrease in sleep latency and an increase in total sleep duration. Nighttime administration in these populations correlates with measurable improvements in daytime behavior and mood. Safety data for these groups is encouraging. Long-term studies tracking patients for an average of 7 years report few and mild side effects that resolve when usage stops. While there has been concern that high melatonin levels might delay puberty, current human data has not established a definitive link to delayed sexual maturation. In these neurodevelopmental contexts, melatonin functions as a chronobiotic regulator that repairs misaligned sleep architecture. While often associated only with sleep, melatonin's broad systemic effects are actually driven by its foundational role deep within the cellular mitochondria. While the pineal gland produces melatonin for circadian signaling, extrapineal tissues like the retina, GI tract, and immune system produce it in much larger quantities. This overview shows the pathways through which melatonin acts as an immune buffer and antioxidant. Within the mitochondria, it provides a direct protective effect by limiting the oxidation of cardiolipin. It also upregulates sirtuin and NRF2 pathways, which are responsible for cellular defense. Simultaneously, melatonin downregulates inflammatory signaling by inhibiting NF-κB. These mechanisms suppress specific inflammatory pathways, including amyloid beta toxicity, nitric oxide release, and COX-2 activation. This activity limits mitochondrial dysfunction, which serves as a molecular driver of cellular aging. Melatonin functions as a regulator of cell death, inflammation, and mitochondrial preservation. This data on anti-inflammatory and amyloid-suppressing activity translates to clinical outcomes in Alzheimer's disease. In aging and neurodegenerative disease, melatonin production in the cerebrospinal fluid and blood decreases significantly. Restoring these levels helps mitigate sundowning, the periods of late-day confusion and agitation common in dementia. Meta-analyses show that melatonin use significantly improves many mental state examination scores in patients with mild Alzheimer's. Researchers are currently investigating these neuroprotective effects for use in cerebral ischemia and persistent post-concussive symptoms. Circulating melatonin levels are also being studied as a potential liquid biomarker to monitor the progression of Alzheimer's. For geriatric cohorts, melatonin addresses the underlying inflammatory neurocascade and disease pathology. Clinicians can build a matrix for melatonin prescribing, separating proven roles from misconceptions. Adult primary insomnia remains in the not recommended category. Strong evidence exists for neurodevelopmental sleep disorders and circadian phase delays. Emerging frontiers include mild cognitive impairment, Alzheimer's sundowning, and anti-inflammatory adjunct therapy. Misclassifying melatonin as a general sleeping pill prevents its use in areas where it has the greatest clinical potential. Clinicians should utilize preparations under 5 milligrams and ensure high-quality sourcing to avoid the variants found in the supplement market. Melatonin is a specialized molecule, not a first-line adult hypnotic. By focusing on the evidence, modern medicine can now utilize melatonin as a targeted chronobiotic and neuroprotective antioxidant.